Stem cells and cloning. I still recall the Sunday afternoon several years ago when a reporter called me at home to seek my opinion about a paper about to be published in a prominent journal reporting the cloning of Dolly the sheep. This was an astounding and unprecedented development, as virtually all scientists, including me, thought that it would be impossible to clone a mammal. Although the entire DNA instruction book of an organism is carried in each cell of the body, it was assumed that irreversible changes in that DNA would make it impossible for an accurate and complete instruction book to be reprogrammed in this way. We were wrong. Indeed, over the course of the last decade, discovery after discovery is revealing the remarkable and completely unanticipated plasticity of mammalian cell types. That, in turn, has led to the current controversy about the potential benefits and risks of this kind of research, characterized by intense public disagreements that show no sign of lessening. The debates about human stem cells, in particular, have been so heated, and the jargon has been so impenetrable, that a bit of background is necessary. A stem cell is one that carries within it the potential to develop into several different types of cells. In the bone marrow, for instance, a stem cell can give rise to red blood cells, white blood cells, bone cells, and even, given the right environment, heart muscle cells. This type of stem cell is commonly referred to as an adult stem cell to distinguish it from one derived from an embryo. The human embryo, formed by union of sperm and egg, begins as a single cell. This cell is phenomenally flexible, possessed of potential to turn into a liver cell, a brain cell, a muscle cell, and every other kind of complex tissue that makes up the 100 trillion cells of the adult human being. The weight of current evidence is that the potential of an embryonic stem cell for sustained replication and the ability to become virtually any cell type exceeds that of an adult stem cell. By definition, however, a human embryonic stem cell can be derived only from an early embryo. Not necessarily at the single-cell stage, but while the embryo is still only a small, compact ball of cells, smaller than a grain of sand. But Dolly was derived neither from an embryonic stem cell nor an adult stem cell. The truly dramatic and unexpected aspect of Dolly's creation is that she came about by a method wholly unprecedented in mammals, one that does not occur in nature. As shown in Figure A.1 in the PDF file, this process, technically known as somatic cell nuclear transfer, SCNT, began with a single cell derived from the udder of a mature sheep, the donor. The nucleus of that cell, carrying the complete DNA of that donor sheep, was then removed and inserted into the rich environment of proteins and signaling molecules found in the cytoplasm of an egg cell. That egg cell had previously had its nucleus completely removed, so it could not provide the needed genetic instructions, only the environment for those instructions to be recognized and carried out. Placed into that primordial embrace, the DNA from the udder cell effectively went back through time, erasing all of the specific changes its DNA packaging had experienced on the way to becoming a very specialized cell involved in milk production. The udder cell nucleus reverted back to its primitive, undifferentiated state. Implanting that cell back into the womb of a sheep then gave rise to Dolly, whose nuclear DNA was identical to that of the original donor sheep. The scientific and medical research world was electrified by the utterly unexpected flexibility of the genome instruction book. Building on that revelation, scientists now see the study of stem cells as a real opportunity to learn how a single cell can become a liver cell, a kidney cell, or a brain cell. Of course, many of these basic questions are being answered by studying stem cells from animals, where the ethical concerns are much more limited. The real excitement about medical benefits from stem cell research, however, is the potential, albeit still unproven, to use this approach to develop new therapies. Many chronic diseases arise because a certain cell type dies prematurely. If your daughter has juvenile onset type 1 diabetes, it is because the cells in her pancreas that normally secrete insulin have undergone an immune attack by the body and have died off. If your father has Parkinson's disease, it is because neurons in a particular part of his brain, the substantia nigra, have died prematurely, resulting in a disruption of the normal circuits that control motor function. If your cousin is on the transplant list for a liver, or a kidney, or a heart, it is because those organs have sustained severe enough damage that they cannot repair themselves. If a means could be found by which we could regenerate these damaged tissues or organs, then many currently progressive and fatal chronic illnesses could be effectively treated, or even cured. For that reason, regenerative medicine is a topic of enormous interest in medical research. At the present time, the study of stem cells seems to offer the greatest promise to realize this dream. A furious social, ethical, and political debate has arisen, however, surrounding the study of human stem cells. The intensity of emotion, the passion of the various perspectives, and the clash of views are almost unprecedented, and often the scientific details have been lost in the storm. First of all, few would argue that the therapeutic use of adult stem cells presents any major new ethical dilemmas. Such cells can be derived from the tissues of an individual who is already living. The desired scenario would then be to convince that cell to morph into the type of cell needed to treat that person's illness. If we knew how, for instance, to convert a few bone marrow stem cells into a very large number of liver cells, then a liver auto-transplant might be accomplished simply by utilizing a patient's own marrow. While there have been some encouraging steps in that direction, and a very significant investment is being made in pursuing adult stem cell research, at the present time we lack confirmatory evidence that the repertoire of adult stem cells that humans possess will be sufficient to meet many of the needs of people with chronic illnesses. Human embryonic stem cells, or alternatively the use of somatic cell nuclear transfer, are therefore being seriously explored as potential alternatives. Stem cells derived from a human embryo should have the ultimate potential to form any type of tissue. After all, they do so naturally in the course of events. But here is where profound ethical questions are being raised, and rightly so. An embryo formed by the union of a human sperm and egg is a potential human life. Deriving stem cells from an embryo generally results in the destruction of the embryo, though a few methods have been proposed that might still allow its survival. If one believes unequivocally that life begins at conception, and that human life is sacred from that very moment onward, then this would be an unacceptable form of research or medical care. Reasonable people will disagree, often passionately, about the appropriateness of such research. Where one comes down on the spectrum of acceptable versus unacceptable is strongly influenced by one's answers to the following questions. Does human life begin at conception? Scientists, philosophers, and theologians have debated for centuries the point at which life actually begins. Deriving more information about the actual anatomical and molecular steps involved in the early development of the human embryo has not really helped with those debates, as this is not really a scientific question. For centuries, different definitions of the beginning of life have been offered by different cultures and faith traditions. And even today, different faiths use different milestones to mark the entrance of the soul into the human fetus. From a biologist's perspective, the steps that follow the union of sperm and egg occur in a highly predictable order, leading to increasing complexity, and with no sharp boundaries between phases. There is therefore no convenient biological dividing line between a human being and an embryonic form that might be called not quite there yet. Some have argued that truly human existence cannot exist without a nervous system, so the fetal development of the primitive streak, the earliest anatomic precursor of the spinal cord, which generally appears at about day 15, could potentially be used as such a marker. Others argue that the potentiality of the embryo to develop a nervous system exists from the moment of conception, and it is not relevant whether or not that potentiality has actually been realized in the formation of any particular anatomic structure. Interesting light has been shed on this issue from the existence of identical twins, who develop from a single fertilized egg. Very early in development, presumably at the two-cell stage, the embryo comes apart, resulting in two distinct embryos with identical DNA sequences. No theologian would argue that identical twins lack souls, or that they share a single soul. In these cases, therefore, the insistence that the spiritual nature of a person is uniquely defined at the very moment of conception encounters a difficulty. Are there any circumstances under which it would be justifiable to derive stem cells from human embryos? Those who feel strongly that human life begins at conception, and that from that very moment the embryo deserves the full moral status of an adult human being, will generally answer no to this question. Their stance would be ethically consistent. However, it must be pointed out that many such individuals have chosen to look the other way, or at least adopt a position of moral relativism, in another circumstance where human embryos are being destroyed. That is the process of in vitro fertilization, IVF, now widely available for infertile couples and widely embraced as a solution to a terrible heartache. In this procedure, eggs are harvested from the mother after a hormonal treatment that results in many eggs being released at once. The eggs are fertilized in a petri dish with the prospective father's sperm. Embryos are observed for 3 to 6 days to assess whether they are developing normally, and then a small number, usually 1 or 2, are implanted into the mother in hopes of achieving a pregnancy. In most instances, there are more embryos available than can safely be implanted. The spare embryos are often frozen. In the United States alone, there are hundreds of thousands of such frozen embryos currently stored in freezers, and that number continues to grow. While actual adoption of these embryos by other couples has resulted in a small number of them giving rise to pregnancies, there is no question that the vast majority of these embryos will ultimately be discarded. A strict stance in opposition to the destruction of human embryos under any circumstance would appear, therefore, to require opposition to in vitro fertilization. A demand that all embryos generated by IVF be implanted has also been proposed, but that would increase the risk of fetal death from multiple pregnancy. There is really no easy way around this dilemma at the present time. Many observers who are otherwise opposed to human embryo research have argued, however, that despite the likely ultimate destruction of excess embryos after IVF, the desire of a couple to have a child is such a strong moral good that it justifies the procedure. That may well be a defensible position. But if so, it challenges the principle that the inevitable destruction of human embryos should be avoided at all costs, no matter what the potential benefits. This circumstance raises the question being asked by many. If procedures could be set up to ensure that no in vitro fertilization procedure was ever undertaken with the explicit intent of generating embryos for research, and if medical research were then restricted only to those embryos that were left over after IVF and clearly destined for destruction, would that be a moral violation?